Hi, my name is Lisa Wagar. I'm an assistant professor in the Department of Physiology and Biophysics, and I'm also a member of the Institute for Immunology and the Vaccine Research and Development Center here at UCI. My lab studies the human immune response to infectious diseases and to vaccines. Currently, there is a crucial gap that exists in our ability to predict what vaccine formulations will actually elicit a protective immune response in people. And so our long-term goal in the lab is to understand and eventually predict what kinds of immune responses are protective against infectious diseases, and then design vaccine formulations that will meet those needs. Historically, people have used animal models to try to understand various aspects of the immune response. And though we've learned a lot from studying mice and non-human primates, for example, in terms of the immunology of infectious diseases and responses, unfortunately, these basic discoveries have not been adequate models to directly translate into understanding human responses. So our goal now in the present day is to try to look at both animal model in vitro data and human samples and try to use all of this information in aggregate to build better models of predicting and correlating vaccine responses with protection. The major site where adaptive immune responses are developed are inside lymphoid tissues, things like lymph nodes, tonsils, spleen, etc. Now, these sites are exquisitely organized into these complicated zones where individual cell types can interact with each other and hopefully eventually build a protective adaptive immune response. Here I'm showing you a germinal center which lives inside lymphoid tissues, and we can see the interactions of various cell types, including stromal cells, follicular dendritic cells, B cells, and T follicular helper cells, all coordinating and interacting with each other. Now, there are several competitive processes that dictate the outcome of these types of interactions. And we can actually see those individual cell types in a real tissue sample interacting with each other. So though we know that lymphoid tissues are the active site where these immune responses are actually developing, we have a very difficult time actually being able to look at these responses in humans. So though we can get access to spleen and lymph nodes from mice, this is a much more challenging problem in humans. So how do we bridge this gap? How does the response in a human versus a mouse compare? And this is the question that my lab is trying to answer. And to do that, we need a controlled system to be able to study the human lymph node response. In my lab, we use something called an organoid to study different concepts of the adaptive immune response. An organoid attempts to recapitulate various aspects of the original tissue, including its architecture and its function. Since we are studying the adaptive immune response, we collect lymphoid tissues. The most accessible lymphoid tissue from a human is tonsils. So lots of people have tonsillectomy for routine purposes, including sleep apnea. So we collect those tissues, we dissociate them into a single cell suspension, and then we can actually cryopreserve those cells for long-term use. When we want to create organoids from that donor sample, we simply thaw the cells and plate them into a transwell plate, as shown here. This transwell enables the cells to remain at high concentration without disrupting the architecture. We stimulate those cultures with a vaccine or an infectious disease of interest, and then over the course of several days, these cells reaggregate and they actually retain some of their original structure. I'm showing you here just a few examples of top-down stereoscope views of organoid cultures, either stimulated with nothing on the left or with a flu vaccine on the right. And you can see that there is a distinct organization that occurs in these organoid cultures. We can use various readouts to be able to understand these vaccine responses that are developing over time and use them to study mechanisms of protection. So we can measure functional readouts of the adaptive immune response, such as antigen-specific antibodies. We can perform mechanistic experiments, which traditionally is quite difficult to do with human samples. So for instance, we can deplete an individual cell type and see what the effect of that cell type is on the ability of the organoid cultures to respond to a vaccine. And we can also directly measure cell-cell interactions and various other aspects of the organization of the cultures using imaging. The Wagar Lab has several future research interests. The first is to try to understand why there is a variability in the individual responses to vaccines. So why is it that some individuals respond more strongly than others? And what kinds of pre-existing histories of those individuals' immune responses might contribute to those effects? We will also have the opportunity to test vaccines and vaccine candidates, as well as potential adjuvants that become available over time. And from a true basic biology perspective, we hope to be able to understand the mechanistic aspects of the immune response through regulating the differentiation of different cell types, altering the immune microenvironment of the lymphoid tissue, and also contributing to the genetic aspects of the adaptive immune response.